So I'm going to start. Everyone can hear me okay? Thumbs up. Awesome. So I'm keeping everyone on mute for now. I'm happy to take questions at the end. Um, so my name's Tal Nuriel. I'm a assistant professor at Columbia University. I've been studying Alzheimer's disease and sort of the role of APOE4 in Alzheimer's disease since about 2013, when I became a postdoc with a woman named Karen Duff, who's sort of a prominent Alzheimer's researcher. Um, I'm not going to be presenting on any of my research, at least not this seminar. I might do that in a future seminar. But for this seminar, I just wanted to sort of um, give what I hope is a very sort of comprehensive and understandable sort of overview of what goes on in the brain during Alzheimer's disease and dementia, and then sort of what sort of the scientifically validated sort of ways to prevent that or reduce your risk. Um, Also, just to note, my former collaborator, Eileen Ruth Torres, is joining us. She was a postdoc in a prominent lab at Weill Cornell. She's now in administrative side, but I asked her to join to sort of help moderate a little bit. So, um, so basically, my talk is, if I can get these things, basically in two parts. First part, understanding Alzheimer's disease and dementia, and then second part, how to protect your brain as you age. So I take this first part especially sort of serious, that I think we sort of hear about Alzheimer's disease and dementia a lot, and we sort of know little snippets, and some people do a lot of research and know a lot. Some people sort of, are brought into this world sort of, involuntarily and don't know anything. Um, and as a scientist, I think we know a lot more, and there's not a lot of communication between the science world and the lay public world. So I want to sort of do what I can to sort of just help people understand at least, what we know or what we think we know is going on in the brain during these diseases. So I'm going to start off with a fairly simple slide, but the it's often even difficult to sort of discern the differences between these, because oftentimes people sort of confuse Alzheimer's disease and dementia, and aren't sure quite what the differences are. And so just as a broad overview, dementia is sort of the umbrella term. Alzheimer's disease is the most common form of dementia. It's the one that we're all sort of most afraid of, probably, or most affected by. And it's the disease that I study. So I am going to be focusing mostly on Alzheimer's disease, but I do want to cover a little bit some of these other forms of dementia: vascular dementia, Lewy body dementia, frontotemporal dementia, or degeneration, however you say it. The other sort of interesting thing, we also sort of characterize uh, we characterize dementia sometimes as loss of memory. That can be the case. It often is the case, but that's not technically the case. Technically, dementia is a deficit, significant deficit, to the point that it impairs daily life of at least two of the following. It can be memory, but it can also be speech and communication, focus and concentration, reasoning and judgment, visual perception. So it's really just two cognitive functions that are sort of impaired. So again, I'm going to be mostly focusing on Alzheimer's disease, and especially for this first part. So to sort of start us off in terms of sort of understanding Alzheimer's disease or the genesis, I'm gonna talk about three women. Two are important to me. One is important to the community at large. But for me personally, I got sort of introduced to this world when I was very young, sort of before I even knew my grandmother, who I called Oma. She was already sort of in decline with Alzheimer's disease. She started, I think, around like 65. And then, lots of visits to the nursing home, and sort of trying to grapple with that, what that meant as a child. And that certainly informs me. And then, my mother, who's still very much alive, still doing well, 78 years old, carries one copy of the ApoE4 gene. But seeing her sort of, having to go through that with her mom and her own fear of Alzheimer's disease, informed me quite a bit. She's currently, and she allows me to share this information, she's currently in the Leqembi prevention trial, and has been for about 4 years. So she's actually sort of out of the placebo phase or placebo or drug phase and is now getting the drug for sure. And then another woman who some of you may know, some of you may not have heard of before, but an important woman in terms of Alzheimer's disease is a woman named August Dieter. Miss Dieter lived in Germany, in Frankfurt, in the turn of the 19th to 20th century. And in her 40s, suffered from dementia. This would probably be called early onset dementia now. Basically reporting memory loss, delusional thinking, trouble sleeping, rapid changes in mood, and generally reported, I had lost myself. So there is one man to introduce, and he actually comes into the picture because of Miss Dieter. He sort of happens to be, her sort of attending physician at the hospital in Frankfurt in 1901, hospital by the lovely name of Asylum for Lunatics and Epileptics, and sees Miss Dieter, takes an interest in her case, diagnoses her. But importantly, in 1906, when she passed away, he had moved on to Munich, but was sent her brain, and did an autopsy, and for the first time saw this sort of pathology that we now sort of characteristically see, but these are original drawings from his 1911 paper, Mr. or Dr. Alzheimer's drawings, basically of plaques and tangles, as he first saw them in Ms. Dieter's brain. So, we now have seen this pathology a million times. You guys are probably familiar with it yourselves. Amyloid plaques and neurofibrillary tangles. The other pathology to sort of introduce is neurodegeneration. So, that's sort of the three common sort of hallmark pathologies of Alzheimer's disease is amyloid plaques, neurofibrillary tangles, and neurodegeneration. So I want to try to sort of zoom in on these in particular, and just because we hear about them a lot, and I just want people to sort of really understand what they are and what they do, and sort of have a better idea of sort of what we're tackling with Alzheimer's disease. So to do that, I'm going to do a little bit of an analogy. So we all know what the brain is. We all know what the brain is one of many organs in each of our bodies, the lungs, the heart, the liver, the brain. So the analogy that I'm going to make for purpose of this presentation is that the human body, one human body, is sort of equivalent to the world, one planet Earth. And in the same way that the human body has lots of different organs, the planet Earth has lots of different countries. And then if you sort of zoom in on one country, for the sake of the fact that I'm in America and a lot of people watching are probably in America, I'm going to use America as the example. Not saying that America is the smartest country in the world, but we're saying that America in this analogy is like the human brain. And then, as with America, you have different regions. You have agricultural regions, you have tech-heavy regions, you have oil-producing regions. It's the same thing with the brain. You have different regions that are sort of responsible for different things, or known to be responsible for different things. So you have the frontal lobe, which sort of codes a lot of sort of personality as well as executive function. So like attention, focus, concentration. And then you have the temporal lobes, which sort of hold a lot of memories, and also a lot of emotional regulation goes on here. Cerebellum is sort of motor control, or motor memory. So you have different regions that sort of do different things. And then if you keep zooming in, so if we zoom in on the medial temporal lobe, sorry, on the temporal lobe into the medial temporal lobe, you get sort of smaller regions, in this case, the hippocampus. For the sake of this analogy, partly because I first presented this talk to a group for my parents who live in Florida, I'm going to say that the hippocampus is like the state of Florida. And then if you keep zooming in, so, every state has lots of different cities. Miami is a, popular city in Florida, big city in Florida. In the same way, if you zoom in on the hippocampus, you're going to get lots of different cell types, the neurons being sort of the most common. And then if you zoom in one more layer, inside every single neuron, you have lots and lots of proteins. You have other things, but for the purpose of this talk, we're going to focus on the proteins. And the analogy I'm making is that the proteins are like the humans in a city. They're sort of the functional unit of the cell. So as is the case with humans, you have lots of, mostly good humans doing good things. You have, doctors and nurses trying to heal people. You have Uber drivers, taking people around. You have garbage disposal people. It's the same thing with proteins. So you have lots of proteins doing actually the same functions. You have proteins that repair things. You have proteins that transport things. You have proteins that dispose of things. So mostly, it's like a city running pretty normally. And most of the proteins doing good things. But unfortunately, as with people, sometimes you get one or two that go bad, or start off good and turn to a life of crime, let's say. For the sake of this talk, because I want to stay away from anything political, I've chosen a cartoon of a 1920s gangster, which is pretty harmless, I think. So basically, the analogy that I'm making is that this gangster is like some bad proteins, and in the case of Alzheimer's disease, the bad proteins to sort of worry about are amyloid beta and tau. And basically, amyloid beta technically is a peptide, but we'll call it a protein for the purpose of this talk. And as with this gangster, maybe alone he wouldn't cause that much problems, but when he gets together with a whole bunch of his friends and they form a gang, they can cause a lot of problems. Same thing with amyloid beta and tau. When amyloid beta or tau sort of aggregates with itself, you get these aggregates, plaques, or tangles, and they can really cause some damage. But, and this is important, the damage that they do isn't really the same. So with plaques, and everyone's heard of, amyloid plaques, and, there's some controversy about whether they're, important or not. My opinion, and I think the consensus in the field, is they are important, but they're not important in that they're the thing that's really driving the worst parts of the disease. So you can have, you can have a brain full of plaques in the same way that you can have a country full of gangsters. And it's not a healthy country. It's not a healthy brain, but life goes on. There are plenty of people who in their hundreds, have a full head of, full brain of plaques and still have relatively good cognitive function. But what happens, or what's thought to happen, is that this process somehow triggers this process. So tau, and this is the basically the analogy that I'm making here, is that tau is like Al Capone. So this is meant to be a cartoon of Al Capone. And Al Capone sort of in the 20s was sort of a more virulent, more violent, form of a gangster that came on the scene and caused a lot of problems. And interestingly, he actually started in Chicago, but then, ended up in Miami of Scarface fame. So the same thing sort of happens with tau. You got these guys doing their thing, causing, some havoc, but the brain's sort of adapting. And then when this process happens, when this tau starts to aggregate, specifically in a region called the entorhinal cortex, that's sort of where it starts to sort of first aggregate. That's when the problems really begin. The entorhinal cortex is a very interesting region. It's sort of known for um, spatial navigation. There's these thing called grid cells that sort of fire off and sort of let you know where you are in a room or in a space. So, one of the common features of Alzheimer's disease is someone might go off driving and forget where they are. And, that's partly because of the sort of the entorhinal cortex starting to have problems. So, if the tau had sort of stayed in the entorhinal cortex, if Al Capone had sort of stayed in Miami, maybe it wouldn't be that big of a deal. But unfortunately, that's not what happens in Alzheimer's disease. You sort of get this spread, or it's not clear if this is going into this and this, or if like new tau is aggregating in this. That's a debate in the field. But you basically have tau that's starting pathology starting at entorhinal cortex, and then eventually sort of moving into the hippocampus, where it really causes some problems in memory. And then you sort of have the tau sort of going throughout the temporal cortex into the frontal cortex. And eventually, as the disease progresses, you end up with the tau sort of throughout the brain, especially in the cortex. And then this is where it gets depressing, so I apologize. What happens is, and it really follows the tau more than the amyloid beta, and I think that's an important distinction that maybe the general public doesn't understand, is that the tau actually builds up inside neurons, as opposed to amyloid that's sort of throughout the brain. The tau is sort of building up within the neurons themselves, and they can kill individual neurons and neighboring neurons. And you basically, during this process, sort of in this same pattern, go from a healthy brain to an atrophic brain. And then unfortunately, that's also where you get the cognitive decline. And there are different stages. Some people say 7 stages, some people say 6, whatever. But essentially, you have sort of preclinical AD, which can be as many as 20 years, where you sort of have some of this pathology going on, especially the amyloid, but you're cognitively totally fine. And then at some point, it can sort of convert into mild cognitive impairment, MCI, which is basically mild but measurable memory loss. So a common sort of test that they might do at a neurologist's office, let's say, is ask you to remember 3 words, and then tell you a story, and then ask you to repeat those 3 words. And that's sort of the short-term memory that sort of starts to go off at this stage. Interestingly, MCI does not mean Alzheimer's disease. It doesn't even necessarily mean amyloid beta. There's also amyloid beta negative Alzheimer's disease, or sorry, MCI. And I think it's around 50-50 people with MCI that develop Alzheimer's and that don't. But unfortunately, many do then develop mild Alzheimer's disease, sort of, further memory loss, maybe getting lost at this point. And then moderate Alzheimer's disease with confusion, changes of personality and behavior, and then severe Alzheimer's disease, which includes loss of the ability to communicate and, needing full-time assistance, which I definitely saw the progression of with my grandmother, which was, not fun. So that's my overview of Alzheimer's disease. Just to finish off this section, I want to sort of go over these other forms of dementia, just so people hopefully understand them a little better. So there's vascular dementia, and vascular dementia is actually much more common, I think, than people realize. It's around a quarter of the dementia cases. And the way I think about vascular dementia anyways is, we think about the brain as looking something like this, but in reality, the brain looks something like this. It's innervated by thousands, maybe millions of small and large blood vessels. And it's very important, that this is sort of a healthy function because the brain is very energy dependent. The brain is 2% of each of our body weight, but it uses around 20% of the oxygen we breathe and around 20 to 25% of the glucose we consume. So the fuel that we get from food is used by the brain. And all of that is delivered through the blood. So if you have issues with blood circulation, cardiovascular disease, different things that cause problems with blood flow, that can, dramatically affect the brain. And then if you have things like strokes, that can seriously affect the brain. One thing to sort of keep in mind here is that unlike these three, which have a somewhat characteristic, or different diseases have somewhat characteristic sort of progression and symptoms, vascular dementia it doesn't function in one way or another. That's because it's really dependent on which parts of the brain are most affected, or where the stroke was, or, the symptoms are going to depend on the part of the brain that was affected. And then I just want to touch on these two, which are lesser known and less common, but I think still important. Lewy body dementia, that's the dementia that Robin Williams had when he passed away, when he committed suicide. And then frontotemporal dementia, sometimes called frontotemporal degeneration. It can be sort of its own disease, but it's also sometimes an umbrella term for diseases like PSP and CBD and MSA, which are all really brutal diseases that hit, in like 40s and 50s. So interestingly, well, interesting to me as a scientist, maybe less interesting as a non-scientist, just sad. You get these, they're basically similar to Alzheimer's disease. They're also proteinopathies, as we call them, where you get this aberrant protein that sort of accumulates out of control and starts causing problems. Alpha-synuclein is the one that sort of goes haywire in Lewy body dementia, forming these Lewy bodies. TDP43 and tau, or tau, I should say. There's usually one or the other for FTD. Although the tau in this case happens in different places than it would have in Alzheimer's disease. But the TDP43 can sort of accumulate and form these TDP43 conclusions. And then again, it's sort of where in the brain it happens that affects the symptoms. So with Lewy body dementia, it can be temporal and can affect memory, but it also affects, like, it causes some weird, like, visual hallucinations. And then frontotemporal dementia, also the temporal lobe, but especially the frontal lobe, which can cause some really awful behavioral changes. And then I'm just going to present this. It's, well, two things. One is it's important to sort of keep in mind that, I can paint a really nice picture of this disease and this disease and this disease, but often biology doesn't work that way. And I'm not a neurologist, I'm a researcher, but what neurologists see in the brain oftentimes is some mixture of the two, or pathologists, I should say. So the most common sort of what they call mixed dementia is Alzheimer's disease and vascular dementia happening at the same time. There's also something called late, which is TDP43 that can often happen with Alzheimer's disease. And sort of the older people are, the more often there's some sort of mixed dementia going on. And then the last one before I move on to the next section is, so I didn't mention two others, but I'm just going to mention them here real quick. These are not technically dementias, which is why I didn't mention them: Parkinson's disease and ALS. But so they're considered movement disorders. but that's not because they don't affect the brain. It's because they affect parts of the brain that sort of play a role in movement. But they're actually very similar to Parkinson's disease and Lewy body dementia are very similarly related. They're both alpha-synuclein. ALS and FTD are very similarly related. They're both TDP-43. But with Parkinson's, it's in the brainstem. With ALS, it's in the motor cortex and the spinal cord. So it's interesting. With the exception of vascular dementia, these are all very similar processes going on, just different regions and different proteins that are sort of going awry. Okay, I'm not taking questions, so I hope that I haven't lost anyone. If anyone is lost, let me know. But I tend to think that people understand more than scientists give them credit for. So I'm going to go into, for the second part of my talk, the next probably 30 minutes, into sort of ways to protect your brain or ways to think about protecting your brain. This is not completely comprehensive. It's not including, maybe some newer, less, studied things, but just the way I think about it, let's say. So if you want to talk about protecting your brain, it's good to understand sort of risk factors, sort of what increases your risk for Alzheimer's disease or dementia in general. So unfortunately, one major risk factor is your DNA, is actually mutations in each of our genes that may increase the risk of, any number of illnesses, as we know from 23andMe and Ancestry, and that can include Alzheimer's disease or dementia. Interestingly, so the way this has been sort of figured out, teased apart, is these things called genome-wide association studies, where essentially you take, a big group of individuals who have Alzheimer's disease, a big group of similarly aged individuals who have, who are not cognitively impaired, and you compare their genes. And we all have millions of different mutations that sometimes are similar to each other and sometimes different, mostly, causing no problems at all. But if we compare all those mutations, millions of mutations, between people who are healthy and people who are less healthy, we start to see things that sort of pop up as being overrepresented in people who are in the, disease group, in this case, Alzheimer's disease. You end up in the case, well, I guess in any case, but for Alzheimer's, this is a very popular sort of plot, the Manhattan plot. And I don't expect anyone to be able to read this, but these are each sort of genes that sort of pop up. And, now we have we're doing these GWAS studies now with hundreds of thousands of people. So we keep getting sort of more and more genes that sort of reach significance as being overrepresented in the Alzheimer's population. And then another way to sort of think about this or visualize this, this is a graph that will be presented at conferences, over and over again if you attend a conference. And what this is basically showing is on the x-axis here, so down here at the bottom line is basically gene mutations, that are either extremely rare or fairly common. And then on the y-axis here is sort of how much that particular gene mutation increases your risk of Alzheimer's disease. So what you might notice is that, I guess we got a drawing on my line. That's all right. What you might notice is that you have a lot of genes that sort of bunch up down here. And what these are basically genes that, are sort of just over that significance line. They've been discovered probably because they're actually pretty common. They are pretty common, 20, 50 more % of the population may have these mutations. but they only increase your risk of Alzheimer's disease, 1%, maybe less. But statistically, they sort of come into play here, but maybe less important to worry about. On the other hand, you have genes like this, APP, presenilin 1, presenilin 2, where the mutations that have been found that are considered to be familial Alzheimer's mutations, also early onset Alzheimer's mutations. And what they found, and they've known for a long time, that if you have a mutation in one of these genes, or a specific mutation in one of these genes, you have basically 100% chance of getting Alzheimer's disease, at least for now. But they're exceedingly rare. Very few people have them. It's less than 1% of total Alzheimer's cases, so not as big of a concern unless you know that this sort of runs in your family. But then what I want to point you to is these two orange circles. One is ApoE4, 1 copy. One is ApoE4, 2 copies. And ApoE4, which is what I study in the lab, is a very interesting gene, I think, in all of genetics, but especially for Alzheimer's disease, because it has sort of the rare and unfortunate distinction of being both a common gene and a gene that impacts your risk of disease pretty significantly. And that's sort of rare in biology, and it probably only exists in this case because it's a disease that happens, or a disease that happens later on in life. Another topic. So I'm not going to talk about, my next seminar I've decided is going to be on Alzheimer's disease, or sorry, on ApoE4 and its role in Alzheimer's disease. So I'm not going to talk about ApoE4 much. But just some highlights. So you have sort of 3 common alleles: ApoE2, ApoE3, ApoE4. Most of us have ApoE3, at least 1 copy. Some of us have 1 copy of ApoE4. Actually, a significant amount of people, almost around 25% of the population actually carries the ApoE4 gene, so it's pretty common. Less, 1 to 2% carry 2 copies of the ApoE4 gene. And as many of you know, if you do carry a copy of, if you do carry ApoE4, it increases your risk around 20%. If you have 1 copy, around 70%. Now there's some debate about whether it might be more than that. If you have 2 copies, another way to think about this is these sort of accelerate Alzheimer's disease. So, people with ApoE3 can get Alzheimer's disease, but these sort of accelerate it. So, and maybe sort of lowers your age of onset, or the average age of onset by around 5 to 10 years per copy. But then the other thing to keep in mind, which I'll go into in my next seminar, these numbers vary quite a bit based on a person's race and ethnicity. So this is mostly having to do with white European descent, but Black, Asian, Hispanic, sort of these numbers change. All right. Hopefully that doesn't stay the whole time, but we'll, I don't know if you guys can see that. So I want to try to erase here. No. All right, I'm gonna leave it. So we also have something called modifiable risk factors. And this is actually maybe a positive thing, or is a positive thing, I should say, that it's not all genes. And this is something that's really, I think really now very strongly appreciated. I think the research has been there for, a couple decades, but we're really starting to take it a lot more seriously, I think, in that there are a lot of different lifestyle factors and different things that can impact your risk of Alzheimer's disease and dementia in general that are sort of modifiable. So the way that I'm going to present this, because just a way that I think is easy to comprehend, and I think very sort of stringent scientifically, is this group called the Lancet Standing Commission on Dementia Prevention, Intervention, and Care. so this is a group that, as the name suggests, the journal Lancet commissioned, I think about a decade ago. And their job is basically to go through the literature and do meta-analyses and different things to try to identify the modifiable risk factors that are most sort of certain or confidently increases in your risk for Alzheimer's disease. And I'm going to go through these one by one, so don't worry if you can't read them. And just to say, this doesn't mean, so if a gene is sort of not on this list, sorry, a gene, if a factor is not on this list, that doesn't mean that it doesn't increase your risk of Alzheimer's disease or dementia. It just means that this group's, sort of very stringent analysis hasn't sort of conclusively defined that, but they put out, in 4 years, they'll probably put out another version that will have a few more that they've sort of decided have sort of met their criteria. And the important thing to realize here is, or to recognize here is what they found is, and what they state is basically that 45% of dementia cases are caused or significantly impacted by one of these modifiable risk factors. And basically what that means is that the, in theory, magic wand, you get rid of all of these modifiable risk factors, you would have 45% fewer dementia cases. So it is a significant sort of impact on dementia, these modifiable risk factors. And I'm going to go through them one by one. Obviously, I can't go through the research on all of them, but just to give you guys an idea. So basically, they're sort of split into early life, midlife, and late life. And this is sort of what the research is showing that, the presence or, lack of presence of these things in these sort of time periods in life sort of increase your later risk of dementia. So in the case of early life, the big thing, and this has been known for a long time, is education. There's a concept known as cognitive reserve that you might have heard of. And this, I apologize, it's sort of elitist, but it's what the research shows, that people who have gone onto higher levels of education, post high school and then graduate school and whatnot, or have more demanding jobs or intellectually demanding jobs, seem to have some protection or at least develop dementia later on in life. That's thought to be because they have some sort of resiliency in their brain that once you have these pathologies and maybe even neurons dying, there seems to be some ability to withstand that for longer. And then most of these you'll notice are midlife. So midlife, 40s to 60s, turns out to be a really important period in terms of your future brain health. So hearing loss is an interesting one that's sort of come onto the scene recently and become more appreciated, that people who have hearing loss that isn't treated, isn't, corrected with hearing aids or what have you, seem to have a later risk of dementia. There's some debate to why that is. Is it the actual signals that aren't going into the brain? Or is it the loss of sort of social connection that comes with hearing loss? That's not fully understood yet. Cholesterol, LDL cholesterol in particular, I'm just going to check if any of these questions are needing my attention. LDL cholesterol, for some reason, has sort of become a little bit, that's a good question. Kimberly asked if the modifiable risk factors have the same impact with people with ApoE4/4. And I think that's a great question. I don't know the research on every single one of them, but a lot of them, certainly like physical exercise and different things, seem to be more impacted by very, very strongly impacted in ApoE4 carriers and even ApoE4 carriers. There was a study recently of Mediterranean diet having the biggest impact in ApoE4 carriers. So ApoE4 carriers seem to be, if they can address some of these, does seem to have a strong effect. Ooh, the line's disappeared. It's good. So cholesterol, that one somehow has become controversial, but I think the research is pretty strong that having high LDL cholesterol in general, during midlife does seem to increase your later risk of dementia. Depression has some interesting correlations with dementia, especially sort of around the time of dementia, but it's also been shown that sort of having untreated depression can increase your risk of dementia later on. Traumatic brain injuries, unfortunately, can increase your risk of dementia later on. Physical activity is an important one. This idea of sort of having a sedentary lifestyle is not great for your brain, especially in midlife. So that's been shown pretty conclusively. So, exercise and, general physical activity is important. Diabetes increases your risk later on. Hypertension increases your risk later on. High blood pressure. Obesity in midlife, unfortunately, increases your risk of dementia later on. And then things like smoking and excessive alcohol use, not surprisingly, increase your risk of dementia. And then a few in late life. One is social isolation. Just sort of this concept that it is very important to stay socially engaged as especially when you're older. Air pollution is an interesting one that's become more appreciated recently, and seems to be able to even accelerate things later on. And then vision loss is sort of similar, I think, to the hearing loss one, that uncorrected vision loss can have some bad impacts, especially later on in life. So,
Well,
yeah,
before you move on, do you want to answer Diane's question about statins in ApoE4 carriers?
sure.
So,
So,
mhm.
yeah, I mean, there's a lot of controversy on this. I mean, from what I've seen, and I've seen some studies that directly measured this, that ApoE4 carriers who had high LDL cholesterol and took statins seemed to benefit quite a bit in terms of dementia, future dementia. So from what I've seen, the answer is yes. Obviously, there's a lot of concern about different side effects with statins, and there are people who I think know these drugs much better than me. I think there's some that maybe don't cross the blood-brain barrier as well. And there's some, other cholesterol lowering drugs that maybe don't have some of the same side effects. So, obviously, if you can lower your cholesterol in a way that is um, that is, going to protect your, other side effects, or reduce other side effects, or protect your brain, you know, I think do that. But I do think, to me, the research shows that lowering your cholesterol is the important factor. But um, I will say I'm not a doctor and certainly not a cardiovascular specialist. So other people may know more than that than me. All right, I'm going to keep going. Um, so so I show this slide, which I think is really interesting. Um, it's not directly related to the modifiable risk factors, but to me, it sort of tells me that, or informs me that our general, sort of community health getting better is having an effect. So this is sort of the rates, the percent of dementia at different ages for people born sort of in successive generations. So this is sort of birth date. And as you can see, there is sort of this, has been for a while, this decline from generation to generation in terms of the percentage of people at that age who have dementia. So basically, we are, as a population, living longer with better cognitive health. the rates of dementia are going up because we are living longer, but in terms of, specific ages, being 60 years old now isn't the same as being 60 years old, 2 generations ago. And I think a lot of that has to do with our sort of collective understanding of different health factors. And then sort of the bad news is, obviously addressing those modifiable risk factors can delay things, but there is always that factor of aging. You do see sort of, this is a study from people who passed away and they did autopsies, and saw sort of more and more of all of these sort of proteinopathies, sort of protein accumulations that were happening in the brains of people who are older and older. So it is, as they say, sort of age is the biggest risk factor for Alzheimer's disease and dementia. So in my mind, that means, therapeutics are important, will always be important, either for people who, just happen to, not be able to address it health-wise, or just they reach a certain age. So I do want to spend most of the rest of the time talking about therapeutics. So this is a chart that I made that is basically all the successful FDA-approved Alzheimer's drugs from 1995 to 2025. And this is not to brag. This is, to me, a very dismal picture, which meaning that this is not, if you compare this to other diseases, this is a very small amount of drugs discovered. There was sort of some early successes in the late 90s, early 2000s with these sort of cholinesterase inhibitors and memantine, Namenda. And these are, still drugs that people take, especially sort of early on. They can sort of improve memory, but they don't seem to sort of slow the course of the disease at all. And then there was just this real sort of wasteland where it was just clinical trial failure after clinical trial failure. And, a lot of pharmaceutical companies didn't even want to touch Alzheimer's because it was, you'd spend money and lose a lot of money. So, that sort of changed. And this, I've been in the field since here. So it really, say what you will about these newer anti-amyloid therapies, they have sort of reinvigorated the field. They're the first sort of successes of recent history, and the first drugs that do sort of slow the actual cognitive decline of Alzheimer's disease. I also added Rexulti, which is Alzheimer's related agitation, just to be comprehensive. But the two that I'm going to focus on here is Leqembi and Kisunla, which most of you know about, but just try to sort of talk about them a little more in depth. So this is hopefully a chart that doesn't scare anyone, and maybe would have scared you, before this talk, but hopefully this is all very clear now. You sort of have this characteristic pattern in the Alzheimer's pathogenesis, where you have, early sort of amyloid plaque buildup, and then tau neurofibrillary tangle buildup, and then brain atrophy, and then memory loss and other cognitive decline. That's sort of, this has been sort of understood for a while. So, maybe not surprising because it is sort of the first pathology, but also I think there's a good argument to be made that way too many resources were devoted to it. The first drugs that we have that are really disease modifying are amyloid. And this was 20 years in the making, really, just to get these drugs, get this process to be successful. And basically what these are is they're antibodies, like we have antibodies for, bacteria and viruses in our own bodies. These are made in a lab and then, injected through infusion into your bloodstream. but they basically target the amyloid plaque in your brain and they clear it out. So this is a diagram, a graph. This is mine, but this one is a graph from the actual Clarity AD clinical trial that looked at lecanemab, uh, and basically what it's showing is that it works really well. It works for the function that it was designed to work for, which is to clear out these amyloid plaques. You can see, in 18 months, really significant amyloid plaque clearance compared to people who are on the placebo. The bad news, or the less good news, is that it doesn't have as significant of effect on cognitive impairment. This is the placebo group, and these were all in this study people who had MCI or dementia, and were on the drug for, I think, 4 years. But this is, maybe I'm lying because this is only showing 18 months. And basically, the people on placebo had a steady decline in their cognition, and the people on lecanemab had a less cognitive decline. So there was a significant improvement, but you can see, these people are still developing Alzheimer's disease. So it's not the cure we want, but it is significant. I've heard this called an additional 1 to 2 independence years, which again is not insignificant. 1 to 2 years of your life is important, but it's not, where we want to be. The reason I sort of still hold out hope for these drugs, and obviously I recommended my mom to go on it, so I'm, maybe biased on it, but so this was Clarity. This was the Leqembi trial. These are the trials that are happening now. The one that my mom's a part of, a lot of people, thousands of people are a part of, these AHEAD 3 and AHEAD 4/5 trials, where they're basically taking people who maybe they have ApoE4, maybe they have a family history of Alzheimer's disease, and they basically test them and find that they have amyloid buildup in their brains, and essentially, but they are cognitively normal. That's the important part. They're cognitively unimpaired. So, if you sort of look at this graph, with a line now, you sort of see that, if you treat people here, which is basically what the first trial did, understandably, because that's how trials work, you treat symptomatic people, you already have a lot of tau going on. You already have a lot of, you already have some atrophy going on. And these processes are probably going to continue, or at least to some degree, without the tau, without the amyloid beta. Whereas I think probably the real purpose of these drugs, or the ideal purpose of these drugs, in my opinion, is to treat before any symptoms develop, while you're still in this sort of climbing phase, and ideally before sort of the tau starts to drive that neurodegeneration. So, that's being tested with Leqembi. It's being tested with Kisunla. Unfortunately, these phase 3 trials take forever. So we won't know the answer until sort of 2027, 2028, whether or not these drugs are doing what people hope they will, which is, delaying or preventing. I think maybe prevention is going to be too pie in the sky, but I do think there's a chance that we wake up in 2027 or 2028 and find out that if given early enough, that these drugs can significantly delay the onset of Alzheimer's disease. But, that's speculation at this point. And then I just want to talk about these other pathologies and how we're trying to target them. So essentially, we have, as I said, sort of the amyloid is kicking off the neurofibrillary tangles. and then the brain atrophy and the memory problems. So, in my opinion, and this is not a novel opinion, the ideal sort of target for Alzheimer's disease is tau, sort of trying to stop this process, which is more sort of the engine of the degeneration. And so that is what people are trying now. Uh, and it's still early days, but not so early that people can't be part of these clinical trials. There's no drugs available, but essentially, as opposed to the amyloid trials that were mostly in phase 3, lots of phase 3s, the tau trials have, a few of them have ended phase 1, where you sort of just look if it's safe in a few individuals, and are now in phase 2, where you're sort of testing different doses and seeing if it has a positive effect. And so I'm listing some, not because it's important to know what they are, but just to sort of give an idea that there are a few. BMS has an antibody towards tau. Johnson Johnson has an antibody towards tau. Eisai has an antibody towards tau. The Eisai one is sort of interesting, at least interesting to me, because Eisai is the same company that makes Leqembi, so it's actually available to people who are on the Leqembi trial, and I think on the prevention trial. I'm not sure if it's only if they develop MCI or what it is, but so I mean, personally, that's interesting to me because maybe for my mother. And it did show some strong effects in their phase 1. It's very small data. It's only a few individuals, but they basically showed 62% at 3 months and 89% at 9 months reduction of tau in the cerebral spinal fluid. So it's doing what it intends to do. Whether it has positive effects on Alzheimer's, I don't know. And then there's this one, B2B, however you say it, 080 from Biogen, which is a little bit different. It's not an antibody. It's like a antisense oligonucleotide, and you have to do interthecal, so it like goes into the spinal cord. Probably not something that a lot of people want to do, but I was actually at the conference where they first showed this data, and this is just from their phase 1 trial, so it's only a couple participants, but basically showing that using tau PET, sort of looking at the tau in the brain of these individuals who got this drug, they saw pretty dramatic reduction, Alzheimer's specific reduction of tau. So, the fact that there are some of these drugs that can do what I think needs to be done to really stop Alzheimer's gives me hope. I wish it was further along, but that said, they're not as readily available as phase 3 trials, but people can get involved with phase 2 clinical trials. And then the last ones, so brain atrophy, they're actually sort of doing a lot with Parkinson's and showing some interesting things, but there is this idea that you can sort of inject stem cells into a brain that already has brain atrophy, already has neuronal loss, and maybe you can sort of replace some of those dying neurons. That's being tested in various clinical trials. Maybe that would have to be done in combination with tau therapy or something, but the idea that you could maybe bring back memory or bring back cognitive function, is an exciting one, but, still very much experimental. And then, my own personal interest, there's all these ApoE4 sort of directed drugs, CRISPR and other things, some which I'm involved in, with the idea that, maybe you can actually correct sort of the things that are going on with the ApoE4 gene or the ApoE4 protein in people with ApoE4, long before they have any issues going on to prevent the increased pathology. And but again, that's still very early. That's not even in phase 1. Actually, there's one that is in phase 1. I'll talk about some ApoE4 targeted therapies in my next seminar. And then I'm just going to finish off with biomarkers. I think I'm about an hour in. just because that I think is a very important topic and one that people are somewhat aware of, but I just want to make sure people are really well aware of. Um, so it used to be the case that people with what they thought was Alzheimer's disease, we didn't necessarily know, oh, they definitively have Alzheimer's disease. Um, and you sort of used to say, this was certainly the case with my grandmother. Oh, we think they have Alzheimer's disease, but we won't know unless we do an autopsy, because the only way to identify it is to look and see if they have these plaques and tangles in their brains. That's no longer the case. It hasn't been the case for 10, 20 years. The real significant sort of development in the past 20 years, I would say, in Alzheimer's disease research is these imaging biomarkers. So the ability to visualize amyloid in the brain, the ability to visualize tau in the brain. We've always been able to visualize degeneration in the brain with MRI. And then the really exciting one, which I think is, would have been a fantasy, 10 years ago, but now is really a reality, is the ability to visualize what's going on in the brain just by looking at someone's blood. And we have a few A beta 42/40 plasma biomarkers. They're good, but they're a little bit unreliable. These p-tau antibodies are more important. NFL is sort of measuring neuronal degeneration. There's also GFAP that measures astrocyte inflammation. But the one I just want to touch on and just make sure people understand is this p-tau 217. So I sort of talked earlier about this process where you sort of have amyloid that sort of probably triggers tau, and then sort of starts to go, towards neurodegeneration. So you don't have to know the biology, but essentially part of this process is something called hyperphosphorylation, where you get lots of these little phosphate groups adding onto the tau protein, and that causes it to misfold and aggregate. So what's been found, which is fascinating and important, is that this p-tau 217, which is basically, amino acid 217 being phosphorylated, seems to be downstream of this amyloid. So what this ends up being a really good marker for is amyloid positivity. So you basically have, so this is sort of an analysis of people who were amyloid plaque negative, people who are amyloid positive, and you can see that it's not definitive, it's not 100% lacking overlap, but it seems to be a really good marker for amyloid positivity. So, but interestingly, it's also sort of like an intermediate between amyloid and tau pathology. So what I think the field is sort of trying to figure out now, this is from 2026, very recent, not even published, but basically just showing sort of like the likelihood of progression to Alzheimer's disease if you have, low p-tau 217 or intermediate p-tau 217 or high p-tau 217. And so I think where we'll get to is sort of like a real understanding of, in the same way of, that we look at cholesterol for heart disease, sort of what a high p-tau 217 level means. And then, hopefully you can like track that and see if you can improve it. Frankly, it's still early days though, for p-tau 217. And then just putting this up, not to advertise, but these are readily available already. Your doctor can prescribe them and figure out what's going on. All right. I'm gonna take a breath. So that is the entire presentation. So I'm going to take questions. I'm putting this up at the end just because I don't know. Part of my motivation for these seminars is to do a little fundraising for the lab. It's a very sort of brutal time in academia with what happened last year, and then just grants aren't being reviewed, and then they're being reviewed in different ways, and a lot of scientists like myself are sort of hanging on by a thread. So I know that I wasn't talking about my research, but raising money for uh, research, and any money that's donated would be going towards directly towards our understanding of what ApoE4 does in the brain, and how to stop it from doing what it does. So I'm just adding that if you're interested. If not, no worries. I think Eileen may put up a link in the chat as well. All right, but I'm gonna leave that up for a second, but I am happy to take questions if anyone has any.
So Tal, we had a bunch of questions, a lot of great engagement. Thank you all for sharing. I think one of the first questions that everyone wants the answer to, that I'm excited to hear, since I also studied APOE, is when are you going to do that APOE seminar next, and how do people find out about it?
Good question. I am thinking March. I have a lot of slides for ApoE4, but I need to sort of put them together and see what else I need to add. But I'm thinking my plan is probably mid to end of March is my goal.
And since everyone joined through the registration link, we'll also have your emails. So Tal will make sure to forward that invitation for the APOE for joining today. So if you're here now, you'll definitely get an email just letting you know that Tal has figured out the date and has that planned.
Yes, true. And I will stop sharing. But thank you all for joining. Yeah, no, go ahead, Phyllis.
The V-Light machine, have you heard of it?
What is it?
V-Light, V-I-E-L-I-G-H-T.
Is that the 40 hertz?
Yes.
I know of the research from Li Wei Tai when she was first presenting it. We were all sort of blown away. It was a little bit almost like this is too good to be true, which either is bad or good, but I think she's, backed it up. Her and I think Ed Boyden at MIT have backed it up, and they've presented a lot on it. And it's this idea that, what they showed in their original papers was that doing 40 hertz, I think the mechanism is sort of that it,
Flashing lights, I have the machine.
yeah, that it sort of activates microglia and sort of maybe causes it to clear out the plaques and do some positive things. I'm in favor of it. It is supported by, it's not, they're doing clinical trials, so it's not, tested to that degree in humans, but the basis of it is strong. There's a little bit of the question of how deep it gets into the brain, but I don't think it does any harm, and I think, or to my knowledge, doesn't do any harm, and I think the science is strong, so I don't have any issues with it.
Okay, thank you.
One of the questions that I don't think you got to in your talk, Tal, that you might speak more about in your next seminar is whether ApoE4 can be potentially treatable by genetic editing such as CRISPR.
Yeah, so I'm planning to sort of give an overview of all of sort of what people are trying in terms of ApoE4. There's one company, Lexio Pharmaceuticals, Lexio Therapeutics, which is headed up by this guy, Ron Crystal at Weill Cornell, where you are. And they're sort of leading the charge here. They've got what they have done is sort of overexpressed ApoE2. What I think they're trying to do now is sort of down-regulating ApoE4 and overexpressing ApoE2 at the same time. I'll talk about this later, the next one, but it is unfortunately, CRISPR gets a lot of press. It's really good at knocking things down, knocking down genes. It's hard to convert one gene to another, and ApoE4 ends up being particularly tricky. So there's definitely a lot of groups sort of working on it and trying to perfect the technology. But it's not ready for prime time, certainly. But I think what may end up happening is this idea that, maybe it's enough to sort of either decrease people's levels of ApoE4 or simultaneously decrease ApoE4 and increase ApoE2. But that certainly is a very active area of research.
And could you give us, since there are many E4 carriers, do you think you can put a positive spin to being an E4 carrier? Are there any positive health effects associated?
That's a good question. Actually, hopefully it's in resubmission right now, but my postdoc Sharita and I wrote a big review about ApoE4 and sort of what it does early on. And there's some reports that maybe there's some increased cognitive function. I think what my opinion on ApoE4 is that what it does well is it sort of allows people to survive on less. So because you're sort of increasing the levels of cholesterol and things like that, they call it sort of a thrifty gene, where you're sort of back in the day when we were roaming the plains or wherever we were and struggling to find food, I think then it had some advantages. I think now, while we're eating Big Macs and having lots and lots of fat and everything else in our diets, I think it ends up having the opposite effect. So, I think I actually do think it sort of is there for a reason, or has stuck around for a reason. But I think in modern society, it's mostly not being that helpful.
Question, what are your thoughts on someone with an ApoE3/4 as it relates to some of those blood tests? Mid-50s, no symptoms. How do you view those tests from a preventative perspective and trying to stay ahead of and keep yourself informed about your own individual progression or potential progression?
Yeah, I think that's a great question. I think that unfortunately, it's a little bit of the Wild West, right? It's like everyone's trying to figure that out on their own, and there's different, groups that are sort of have their own analyses going. But there's no sort of like doctor recommended guidelines, which I sort of wish they would. I mean, I get it, the research isn't definitive yet, but I still sort of feel like some guidance on that would be helpful. My opinion is, so full disclosure, have 2 copies of ApoE3 and I'm 46 years old. So I'm not checking myself yet. If I had a copy of ApoE4 and was mid-50s, I would probably start. I think probably in your 50s, in my opinion, is probably the earliest you need to, and even that's early. But I do think it offers a real ability to sort of self-monitor where you're at. We don't know enough about sort of what that means long term and whether you can reverse it and whatnot. But I think it does give you the opportunity to sort of see what's going on in your brain, and hopefully the levels are low, and maybe gives you a little bit of peace of mind.
Thank you.
Happy to take any other questions, but no requirement.
Thank you so much, Dr. Nuriel. I'm curious, there's such conflicting information about statins. It's hard to know who to trust, because everyone has an angle and they're making so much money on these statins. And some say they even cause diabetes and dementia and liver problems. And I'm just so torn. I'm taking them, I hate taking them, but I'm wondering, are they really necessary if I can curb my diet and intake?
Well, and I think that sort of gets to it, right? And again, I'm not a doctor. I don't want to make, I'm a PhD, but I didn't go to medical school. So I don't want to make any medical recommendations specifically, but I mean, I think that's sort of the thing, right? I mean, to me, I think there's this sort of idea out there that maybe, the cholesterol being bad is a hoax. And I mean, I think there's some truth to like maybe what we consume, the cholesterol we consume in our diet isn't as important, but I do think having high levels of cholesterol does, put you at risk for, heart disease and dementia. So I think the goal is to lower your cholesterol levels. If you're able to do that, without statins, I think that's better. I mean, who wants to be on any medication? I think if you're not, again, I don't know, maybe this is something I need to sort of do my own sort of looking into and decide on for myself. But I mean, there may be other ones that are sort of better than some, but I don't know. If it were me personally, I think I would, and I think both my parents are on statins, so I would prioritize sort of heart health. And then, but yeah, like you said, I think there's some possibility of, pre-diabetes or different things that you have to sort of watch out for. But I don't know. I'm part of mainstream science and medicine, I suppose. So I tend not to be too hard on that stuff.
Can I ask Tal, maybe
Sure.
kind of plug in my own personal interest? Do you think you'll cover sex differences in a future seminar, especially related to, some of the drug trials like Leqembi?
That is a great question. I
Or
mean,
maybe I
certainly
can maybe I can plug a recommendation for some other scientists to give that talk with you.
fair. No, well, I think it's important. I mean, one of the biggest questions is this sort of hormone replacement therapy.
Mhm.
And that's another one that's hard to say because there's some conflicting data out there. But it's certainly with ApoE4, it's a very important consideration, which I will include. But yeah, it might actually be, I think you're right. It might be good to pull in someone who is a little more of an expert on the sex differences, or maybe do another seminar on that. All right. I think we're reaching the end of the interesting questions. So, all right.
You've got one more from Betty. Is there any correlation between the shingles vaccine and reducing AD?
Seems to be. That's another very interesting one. That could be its own talk too. The sort of effects of bacteria and viruses on AD pathogenesis. There's a lot of research on it, and that's pretty convincing. The rates of dementia sort of reducing after the sort of implementation of the shingles vaccine. And there does seem to be a few different sort of viral mechanisms that sort of accelerate Alzheimer's disease. So I actually believe that one. Yeah. All right. Thank you very much for joining. I appreciate it. And I'm gonna hopefully this recorded properly, and I'm gonna put it up and other people can watch as well. But appreciate you guys spending time with me.
Thank you.
All right.
Thank you.
Have a good day. Thanks, Eileen.
You're welcome.
The recording has